Hi everyone, I'm Xiao from CSBL. Today, my presentation title for the Journal Club is Integrated Meta-omics Approaches Reveal Sccharopolyspora as the core functional genus in huangjiu fermentation, which published on biofilms and microbiomes on September this year. Here are the highlights of this journal. In this study, metagenomics, metabolomics, microbiome isolation, and co-fermentation were used to investigate huangjiu. Metagenomic analysis in this study showed that bacteria Sccharopolyspora are predominant in vat tree and responsible for breakdown of starch and cellulose. Metabolic network and correlation analysis showed that Sccharopolyspora rectivigula, Sccharopolyspora erythrea, and Sccharopolyspora hirsuta made the greatest contributions to huangjiu's metabolites, consisting of alcohols, esters, amino acids, and organic acids. Sccharopolyspora hirsuta J2 isolated from vat tree had the highest amylase, glucoamylase, and protease activities. And co-fermentations of Sccharopolyspora hirsuta J2 with Saccharomyces cerevisiae HJ resulted in a higher fermentation rate and alcohol content, and huangjiu flavors were more similar to that of traditional huangjiu compared to co-fermentations of Aspergillus or Lactobacillus with Saccharomyces cerevisiae HJ. And genome of Sccharopolyspora hirsuta J2 contained genes encoding biogenic amine degradation enzymes. Huangjiu is a type of Chinese alcoholic beverage. Archaeological records suggest this beverage has been made for nearly 9,000 years. Huangjiu is brewed by mixing boiled grains, including rice, glutinous rice, or billet with qi as a starter culture, followed by saccharification and fermentation at around 13 to 18 degrees for fortnights. Its alcohol content is typically 8 to 20%. Here I'm going to introduce fermentation process of vat tree and huangjiu. For vat tree production process, first the vat is crushed, blended with 20% water, and then pressed into molds of different size depending on the manufacturer, which are called vat tree blocks. And then the vat tree blocks are moved into tree house for culturing under ventilated and insulated conditions. Fermentation is divided into 3 fermentation stages. First, 3 to 40 degrees for 3 days, which results in the mass reproduction and growth of microorganisms. Second, 45 to 55 degrees for 4 to 5 days, which is used mainly for the accumulation of microbiome metabolites. Third, 10 to 12 days above 40 degrees, which is conducive to moisture removal in the starter core and the formation of aroma substances. The vat tree will drive to a water content below 12% weight. After cooling down at room temperature, the vat tree was stored for 60 to 90 days. For huangjiu production process, first the rice is washed and soaked in the natural water at room temperature for 40 hours, and the soaked rice is steam cooked in a steaming machine to gelatinize the starch contained in the rice grains. Then the steamed rice is mixed with vat tree, water, and Saccharomyces cerevisiae, and the mixture is called huangjiu fermentation mash. This process can be divided into 2 stages. The stage 1 fermentation temperature is 28 to 30 degrees and lasts for 3 to 4 days, and stage 2 lasts 15 to 20 days and the temperature controlled below 15 degrees. After fermentation, the huangjiu fermentation mash were filtered by pressing, sterilize, and store the fresh huangjiu. To uncover the variation curve of microorganisms during fermentation, microbial DNA copy numbers and community structure were analyzed during quantitative real-time PCR and metagenomic analysis. Metagenome sequencing showed a total of 403 genera and 1,442 species in the wet tree sample obtained from a commercial huangjiu factory. Bacteria were dominant, constituting 90.24% of the microorganisms present and were predominantly in the Actinobacteria phylum. Only 6 genera were detected at larger than 1% abundance, and Sccharopolyspora and Streptomyces were the most represented. Other taxa included Bacillus, Pseudomonas, Amycoleptosis, and Lichthymia. Metagenome sequences were also able to resolve species-level differences. Sccharopolyspora hirsuta was the most abundant, which is 38.8%, and followed by other species of Sccharopolyspora. And here the results shows dynamics of functional microorganism during huangjiu fermentation process at species level. At the 25, 50, 100, and 300 hour time points during the fermentation, the microbial contents of the huangjiu fermentation mash were mainly comprised of Actinobacteria, Firmicutes, and Ascomycota phyla. The relative abundance of the dominant genus, Sccharopolyspora, increased from about 40% to 50% during the huangjiu fermentation process. A variety of Sccharopolyspora species were detected, and Saccharomyces, specifically Cerevisia, was also present in the huangjiu fermentation mash, and reached the highest levels, about 22% at 100 hour post incubation, and then declined to 11%. Aspergillus was also present in more modest amounts, about 10% after 25-hour incubation, but declined over time to 4% of the total micro contents by 300 hours. The abundant metabolites were the most outstanding feature of huangjiu, comparing to other fermented alcoholic beverage. So in this study, authors characterized metabolites during the fermentation process, and a total of 103 metabolites were identified in the wet tree sample tested, including 8 organic acids, 18 amino acids, and 77 volatile compounds. Metabolites present in the huangjiu fermentation mash changed over time. Lactate, tartrate, and citrate were the dominant organic acids in huangjiu fermentation mash. 7 amino acids accounted for between 58.4% at 400 hour to 70.2% at 100 hour of the 18 amino acids detected. 64 volatile compounds were also identified in the huangjiu fermentation mash. 5 alcohols, 10 esters, and 3 phenols were the main volatile compounds of huangjiu fermentation mash. Among the identified volatile compounds, the aroma compounds were the most abundant. To explore the correlation between the microbiota and main metabolites, O2PLS method was used to analyze the change in the relative abundance of core functional microorganisms and main flavor metabolites during the fermentation of huangjiu fermentation mash over time to explore the correlation between the microbiota and the main flavor metabolites. As shown in the figure, the proportions of 5 microbial species, including 4 bacterial and 1 fungal species, had a significant influence on the flavor metabolites. Among them, the O2PLS model showed that Sccharopolyspora was the most important contributor to the production of flavor metabolites during huangjiu fermentation. According to previous study, researchers co-fermented Ashercita J2, A. flavus SU16, A. auris MQ, and A. platyrum JN01 with Saccharomyces cerevisiae respectively and profiled metabolite changes during fermentation. This slide shows the main physicochemical results of the huangjiu according to the national standards of China. All the final fermentation products met the national standards except for huangjiu inoculated with platyrum JN01. Fermentations with Ashercita J2 had the highest fermentation rate and final alcohol content than others. Similarly, the reducing sugar content of the Ashercita J2 group remained low during the fermentation and was significantly lower than that of control. Compared with the control group, the speed of Aspergillus group's fermentations was slightly higher. However, the final alcohol contents were not significantly different with the control group. Sugar in Aspergillus group was exhausted till at 300 hours, and their value were not significantly different from these of the control groups. The fermentation inoculated with L-platyrum JN01 did not result in ethanol production, but the reducing sugar content increased first and then decreased as the fermentation proceeds. The titratable acid content of the L-platinum JN01 group also increased rapidly, and samples showed obvious rancidity. The titratable acid content of the other groups increased with fermentation, but remained within a reasonable and stable range. By the end of fermentation, the amino nitrogen content of each sample meets the superior standard of huangjiu. The different metabolites of huangjiu fermented with Ashercita J2 had similar proportions compared to those in the control group. Hierarchical cluster analysis showed similarities between Ashercita J2 and the control group based on their main metabolites. These results indicate that the metabolite profile of traditional huangjiu could be simulated by co-fermentation of Ashercita J2 and S. cerevisiae. By comparison, concentration of metabolites was similar between huangjiu containing the A. flavus SU16 and A. auris MQ inoculants, but not to the controls. The Ashercita J2 genome encodes genes required for BA's degradation, according to the CAC pathway analysis. BA means biogenic amine. It's a biogenic substance with one or more amine group. They are basic nitrogenous compounds formed mainly by decarboxylation of amino acids and are formed during fermentation. So, these results indicate that Ashercita J2 has the potential to degrade BAs. So here, researchers tested the BA degradation capacity of Ashercita and two Aspergillus bacteria tested for co-fermentation previously. After the fermentation, the concentration of total BAs in huangjiu inoculated with Ashercita J2 was significantly lower than huangjiu inoculated with others and control group. To further explore the capacity of Ashercita J2 to degrade BAs in fermented foods, Ashercita J2 was inoculated into sausages and soy sauce for fermentation. The pH, moisture content, crude protein content, and soluble solid, total nitrogen and amino nitrogen content in soy sauce and sausage made with Ashercita S2 were not significantly different from the controls. At the end of fermentation, BA concentration in the soy sauce and the sausage was lower when Ashercita J2 was added to the ferment, constituting a significant decrease decreased total BAs approximately 40 and 60% compared with the control group in sausage and soy sauce fermentation respectively. In this study, metagenome sequencing, metabolomics, strain isolation, and characterization were combined to identify the core microbial taxa in huangjiu fermentations and the corresponding metabolites that they make. Sccharopolyspora is the dominant member of the wet tree and huangjiu fermentation mash microbiota. Specifically, it showed that Sccharopolyspora, a genus of bacteria that has thus far only been associated with the production of bioactive natural products, is a key member of the huangjiu fermentation. A researcher's findings also showed that huangjiu fermentations are dominated by bacteria, not fungi. Isolation and characterization of several Sccharopolyspora strains showed that these bacteria possess enzyme production capacities. Last but not least, researchers found that Sccharopolyspora Herceta J2 can also degrade BAs. This is the end of my presentation. Thank you for your listening.